Good morning, everybody. Thanks for joining us today. I'm Emily Silva. I'm the NECAN coordinator. Welcome to the 2023 NECAN webinar series. This is the third webinar in this series as we collect our monitoring priorities for the Northeast. And today we'll be focusing on OA and climate. With the assistance of this series and our presenters, the NECAN Steering Committee will be working on the development of a regional monitoring plan for OA, and these webinars will serve as an informational resource for them as the plan begins to come together. Updates on this webinar series will be shared through our mailing list, but can also be found on the NECAN website at the link listed below on my slide. I'll also put it in the chat. At the conclusion of the presentations today, the steering committee will be asking our panel some questions, and then we'll open it up to a more general Q&A for the rest of the attendees. Please feel free to use the raise hand function on the bottom of your screen if you have a question, or you can submit questions and comments through the chat. First up, we'll hear from Xinyu Li. Xinyu is a PhD candidate at the University of Delaware, working under the guidance of Dr. Weijun Kai. Her research is primarily focused on the carbon cycle of coastal regions with a specific interest in seawater carbonate chemistry, spectrophotometric pH measurement, and anthropogenic carbon quantification and its impact on coastal acidification. In response to monitoring ocean acidification and long-term seawater carbonate chemistry changes, the NOAA Ocean Acidification Program sponsored several comprehensive coastal cruises in recent decades. Xinyu will be talking about the carbonate data set from 1996 to 2018 from these synoptic cruises in the Mid-Atlantic Bight to examine coastal anthropogenic carbon accumulation rates, mechanisms, and potential influences on the coastal region. I'll hand it over to you, Xinyu. You should be able to share your screen.
All right, thank you. Let me share my screen first. All right, can you hear me and see my screen okay?
Yep, everything looks great.
Okay, great. Hello, everyone. My name is Xinyu. I'm from the University of Delaware, working with Dr. Weijun Kai. I'm a PhD student that will graduate this summer. And thank you so much for giving me the chance to share my work with all of you today. And today I'm going to talk about anthropogenic carbon estimation from the US East Coast OCA acidification cruises. So before I start my content, I would like to acknowledge all the scientists and the crew members that help us during the cruise, especially the PIs of this cruise, Dr. Joe Salisbury from University of New Hampshire, Dr. Rick Vannihoff from NOAA AOML, and Dr. Weijun Kai, my advisor from University of Delaware. And also, I would like to thank all the scientists from different institutions that help us on board. That includes Columbia University, University of Connecticut, and the University of Miami. Dr. Chris Landon's group help us to measure the dissolved oxygen. And especially, we would like to thank the NOAA Ocean Acidification Program that sponsored all the East Coast acidification cruise and also my PhD work. And of course, the captains and the crew members on board that help us to operate CDD and the instrument, and my lab colleagues. And without them, it is impossible to achieve such a high quality carbonate parameter measurements. Okay, let's get started to the content. So since the Industrial Revolution, the atmospheric CO2 concentration is become higher and higher dramatically. And the current atmospheric CO2 concentration is much higher than the oscillation range over the past 800,000 years ago. And it leads to the global temperature increase and climate change. But luckily, not really luckily, but the rising atmospheric CO2 is tempered by the oceanic uptake. The ocean uptakes a lot of CO2, and we have lots of research going on to assess the relative role of the ocean as a sink for anthropogenic carbon through a lot of global observational-based research and also from lots of modeling work. And as the ocean absorbs lots of CO2, the seawater is become more acidified and the seawater pH drops. The absorbed CO2 consumes the carbonate ion and impedes the calcification of the shells. So it leads to another problem, acidification. So in order to monitor the coastal ocean acidification, the NOAA OAP program supported a lot of coastal cruises. On the East Coast, we have East Coast Ocean Acidification cruises that covers the majority of the East Coast of the United States and also some Canadian coast. And the East Coast Ocean Acidification cruises were conducted in 2015 with research vessel Golden Gunter, and 2018 on the research vessel Henry Bigelow, and 2020 summer with the research vessel Ronald H. Brown. That just happened. The goal of these cruises are to provide synoptic characterizations of carbonate parameters and to provide climate quality OA data that can link to the global ocean acidification network. And it also provides a very high quality and long-term record of data to help us to explain the dynamics and the controlling process of the ocean acidification. Next, I'm just going to go a little bit into more details about the ECOA cruises. And during the cruise, we have lots of station operations. We have CTD cast for us to sampling from the NECAN bottle directly. And we have AOP, IOP profile cast. We have lander cast for bottom waters. And we also have some bungo net cast for biology. And we also do some underway operations. We have underway instruments to measure total alkalinity, pH, PCO2, and oxygen-argon, that is oxygen-argon ratio, that is an indicator for the net community production. And during the underway process, we also collect underway flow bottle samples. That's to help to calibrate the underway instrument measurements. And the most important, we have very high quality carbonate data measured. Our lab in University of Delaware, we are responsible for the total alkalinity and the spectrophotometric pH measurements, and the AOML are responsible for the dissolved inorganic carbon measurements. All of these measurements following the guide to the best practice for the ocean CO2 measurements. Those parameters are measured on board. And also we are sampling the nutrients, the particular organic carbon, the dissolved organic carbon, and the dissolved inorganic carbon, carbon-13 isotope, to bring those samples back to measure in the lab. So about the cruise data quality, all the ECOA cruises data, the 2015 and the 2018 were archived in the coastal ocean data analysis product, the North Atlantic part. And as you can see here, the East Coast cruises are flagged as cruise flag B. This indicates they are the dedicated ocean acidification cruise with very high quality carbonate parameter measurements, though it may not have as much as deep stations, that is the deeper than 2,000 meter and 2,500 meters as the cruise flag A data. But this is improved in the past ECOA 3 that just happened last year because we have more deep stations. And all the data qualities here met the climate quality of pH measurements. So what we learned so far from the ECOA cruises, we have conducted the synoptic spatial analysis of the carbonate parameters. This is published by my advisor 2 years ago. We reviewed the meridional distributions of the DIC, TA, and pH, and omega. Omega is carbonate saturation state. It is an indicator to show the carbonate dissolution. If omega is smaller than 1, the carbonate begin to dissolve in the seawater. So let's focus on the figures here. From the top to the bottom, they are the surface distribution patterns of DIC, total alkalinity, pH, and omega. So for the pH patterns, it is high in the south part and low in the north. It is closely related to the seawater properties, and the distribution of total alkalinity is physical mixing process dominated. As for the DIC here and the omega, they also have a meridional trend here, but DIC and omega are mainly dominated by the air-sea gas exchange process superimposed on the physical mixing. That is, if we calculated the equilibrated DIC, which we calculated from the seawater total alkalinity and the atmospheric PCO2, we can get a very similar pattern of equilibrated DIC, and actually we observed the DIC. But for the pH, we cannot really see an obvious pattern of the pH. This is because pH is mainly dominated by the local and biological process. The air-sea gas exchange process and also the physical mixing process, they are kind of counterbalance each other, so make the local and biological process more clear. And this is about that was about the surface, and now we have a quick look at the vertical distributions. Still, the 4 parameters we focused on here is from the top to the bottom, there are TA, DIC, pH, and omega. And for the total alkalinity and the DIC, we see they generally increase with the depth, but their reasoning are different. For the total alkalinity, it's still more like represents the physical mixing conditions, but for the DIC, it's more complex. It includes the physical mixing and also the biological influences here. And for the omega here, we see omega decrease. Generally, omega decrease with the depth. But when we concentrate on the pH vertical distributions, the pH concentration is not increasing with depth. Actually, we have a pH minimum layer in the depth around 500 meters. This is in the south part of the East Coast, South Atlantic Bight, and it moves to a shallower depth in the Mid-Atlantic Bight and also the slope water outside of the Gulf of Maine. And this low pH layers here is mainly because of the organic matter remineralizations. And this, what I talk about is the surface and the subsurface spatial variations. And the next, I'll begin to talk about the temporal analysis of the carbonate parameter. And now we only focus on the Mid-Atlantic Bight region. And in the Mid-Atlantic Bight region, here is a Long Island Sound. Here we call it New Jersey Transect. This is a Delaware Bay, and this is a Delaware Transect, and then this is a Chesapeake Bay, the Chesapeake Bay Transect, and this is the Massachusetts Transect, which is also called Line W in some global ocean surveys. And we see from the Massachusetts Transect, which is the most north transect here, we see the water column DIC concentration is increasing. The color is becoming redder and redder with years. And also, if we compare the pH distributions between the two cruises, here I use the GOMAC 2 cruise, the 2012, and the ECOLA 2 cruise 2018 as a comparison. The reason I choose those two time frame is the GOMAC 2 is the starting point where we have very high quality spectrophotometric pH measurement. So it can detect very small changes. So this comparison in the Massachusetts transect and the Chesapeake Bay transect, the most north and the most south transect in the MAB, it clearly reveals a pH decrease and in a magnitude about 0.01 to 0.015 from 2012 and 2018. So only from the observational data, we see both the DIC decrease and the pH decrease. But still, it is very hard to tell whether it's because of anthropogenic influences or the rising CO2, or it's just a natural variation. So here we conducted a research and to investigate the anthropogenic carbon accumulation. Here in this research, the question we try to answer is how much anthropogenic carbon is accumulated in the coastal region. Excuse me. We mainly focus on the mid-Atlantic Bight region, and this region is influenced by the southward Labrador coast current, which carries cold and relatively fresh water from the north. And on the offshore part, it is influenced mainly by the Gulf Stream water that mainly carries the high temperature and high salinity waters. And we also have some estuarine inputs from large estuaries like Chesapeake Bay and Delaware Bay. And this is the regional setups in the MAB. And for the coastal cruises we are using, we not except the two ECOLA cruises, we also use the two GOMAC cruises that in 2007 and in 2012, and the earliest available carbonate measurement back in 1996. So we took advantages of this 22-year high quality carbonate data since 1996, and we used a method called extended multiple linear regression method, combining a time series analysis to assess the anthropogenic carbon accumulation during this period. And this method is widely used in the global oceans to estimate anthropogenic carbon, but it is rare to be used in the coastal ocean because the first reason we don't have long enough time series to detect the change in the coastal region, and the second reason is the noise to the signal ratio in the coastal region is relatively large. And then comes to the results of this anthropogenic carbon analysis. I would like everybody to focus on the slope water first, and we can clearly see a sandwich structure of the slope water anthropogenic carbon distribution. On the top 200 meters, we have the highest anthropogenic carbon accumulation from 1996 to 2018, and it increased about 25 to 30 micromole per kilo, and it is around 1.2 micromole per kilo per year. And these are the Gulf Stream water. And in the middle, we have a very low anthropogenic carbon accumulation that is smaller than 8 micromole per kilo in 22 years. And this is water from Antarctic intermediate water plus some other intermediate waters. It has a feature of high AOU and low oxygen. It also has low pH, relatively old water. And in the bottom, below the 500 meters, we have the upper layer of the North Atlantic deep water. And some people also call it the Labrador seawater. And in this water mass, we have anthropogenic carbon accumulation. It's about 15 micromole per kilo. in the 22 years. That is about 0.6 micromole per kilo per year. And if we see the feature from the offshore to the nearshore, even here is not really clear, but the offshore anthropogenic carbon accumulation is higher than the nearshore. So come to explaining the controlling factors why we have this kind of sandwich structure of anthropogenic carbon accumulations. I summarize them to two factors. The first one is the water age. So for the Gulf Stream waters, it has a relatively young water age because it flows very fast, very fast speed from south to the north. And for the North Atlantic deep water, it is formed by the winter convection in the Labrador Sea region here, and it takes about 20 years to sink from the surface of the Labrador Sea and flow along the coast to the Mid-Atlantic Bight region here. So actually, the anthropogenic carbon we quantified in the North Atlantic deep water, it's a reflection of the Labrador Sea surface water absorbing CO2 back in 20 years ago. And at that time, from 1970s to 1990s, the atmospheric CO2 increase is much slower than the most recent 22 years. And for in the middle, it is the Atlantic intermediate water, which is originated from the Antarctica. The age of this water is very old, and it travels a very long time to the Mid-Atlantic Bight. So it naturally contains less anthropogenic carbon. Overall, the first factor is older water has less anthropogenic carbon. And the second factor is the water properties at the surface. So the Gulf Stream water is mainly, I call it the Gulf Stream water, but actually it's mainly composed of the North Atlantic Central water formed here. And the water properties at the surface is usually high temperature, high salinity, and the high total alkalinity. And for the North Atlantic deep water, when it was at the surface, it has the property of low temperature and relatively low salinity compared to the Gulf Stream water, and also relatively low total alkalinity. So we conducted a very simple simulation to show that the low buffer capacity water absorbs less anthropogenic carbon. So these are the two factors that leads to the sandwich structures of the anthropogenic carbon on the slope in the Mid-Atlantic Bight. And what about other OA matrices respond to the anthropogenic carbon? So we do a further calculation. Here, figure on the top left is the anthropogenic carbon accumulation since 1996, and the top right figure is the corresponding pH change during these 22 years. So as we can see here, the Gulf Stream water, because it has the most anthropogenic carbon, it also leads to the most anthropogenic pH changes. Overall, the anthropogenic pH change rate is about 0.01 to 0.02 per year. So the observed pH decrease is almost the same rate as the anthropogenic pH change rate. And also, if we look at the Wiley factor, the Wiley factor is a factor to describe the seawater buffer the sensitivity. Even though the North Atlantic deep water does have less anthropogenic carbon compared to the Gulf Stream water, but in terms of the sensitivity change, it is almost the same as the Gulf Stream water. So this indicates that the low buffer capacity water is more sensitive to the anthropogenic carbon accumulation. And this can also be verified by we compared the anthropogenic pH ratio to the anthropogenic carbon ratio. The North Atlantic deep water has higher anthropogenic pH to anthropogenic carbon ratio. This means even the same amount of anthropogenic carbon accumulation in the water, the North Atlantic water's pH gonna change more because of the anthropogenic carbon accumulation. And last, this is not like the results, but more like an extension from my research. I'm going to talk about the influences of the anthropogenic carbon on the continental shelf water. And as we present earlier, the anthropogenic carbon offshore is higher than the near shore. So and also we plot the anthropogenic carbon against the salinity. Anthropogenic carbon is relatively a good conservative parameter in the subsurface because it does not change anymore when it is isolated from the air. So the mixing with the water, the offshore water with high anthropogenic carbon to the near shore can have a potential influence on the continental shelf water. And especially if you are familiar with this region, we have a cold pool here between the Massachusetts transect to the Chesapeake Bay transect in the summer. So it is our interest to study how it is influenced on the low pH and low buffer capacity near shore water. And another possible research interest from the anthropogenic carbon is actually in the Gulf of Maine region. Here is a map of Gulf of Maine, and this transect is especially important because it has the northeastern channel. It cut across the two channels, and the first one is the northern channel, and the second one is the northeastern channel. Based on previous research, the slope water gets into the Gulf of Maine through this channel, and especially the slope water sit in the bottom of the Gulf of Maine region. And the flowing slope water can be either the warm slope water that is adjunct to the Gulf Stream, which naturally have a very low pH, or sometimes sometimes it can be the Labrador Sea water. And in my definition, it is the upper North Atlantic deep water. If you still remember, it's the water contains relatively high anthropogenic carbon. So in the future research, it is also our interest to see how the anthropogenic carbon, high anthropogenic carbon slope water intrude to the bottom of the Gulf of Maine through the northeastern channel. How does it influence the carbonate chemistry and the future, even the ecosystem influences on the whole region of the Gulf of Maine. So for the future monitoring strategies, so far, we already have a very comprehensive, high quality and ocean acidification focused research course. And it already supported a very systematic analysis of the carbonate chemistry in the whole East Coast region. This is something we have already achieved. And also we added lots of deep stations and offshore stations in the Northeast that would be able to connect the coastal region better with global ocean observations and to make these two products more comparable, two kind of cruises more comparable. And especially it will benefit from have more offshore stations to track the impact of the Gulf Stream on the whole coastal regions. And also we added additional transects on the north of the Nova Scotia and would help further to investigate the influence of the water from the subpolar region on the Gulf of Maine. And in the future, I feel it's very important to continue this kind of climate ecology, climate quality OA cruises, because I can do the anthropogenic carbon estimation in the MAB is because we have long enough high quality data measurements. And in the SAB, we already, we begin to have this capacity, because from the GOMAC 1, which happened in 2007, and to the ECOA 3, which happened in last year. So it's already 15 years time gap with time differences. So we begin to see the signal of the anthropogenic carbon a little bit more clear. But for the Gulf of Maine region, we still lack of this kind of data accumulation. And the fifth, I would suggest to have more observations to support the exploration of the temporal evolution of the cold pool regions, which is very important, have some economic importance, because it is a home for some economically important species, especially in the summer. And we already added a couple more stations, but in order to fully understand the cold pool, I would strongly suggest to have some surveys from winter seasons, because the cold pool is formed it's originally the winter remains water at the surface, and then it's gradually trapped in the subsurface by the thermocline. So it is important to know from the beginning to the end. Okay, the last page is my take-home message. In this presentation, I briefly introduced about the ECOA cruises, and this comprehensive ECOA cruises support synthesis of the carbonate system, especially to explore its controlling factors and the change under the climate change. The surface DIC and omega is mainly dominated by the partial equilibrium with the atmosphere superimposed on the physical mixing. And the third point, and also the most important point in this presentation, is the anthropogenic carbon accumulation. So the slope water is mainly controlled by the water age and the properties. And also, I think for the future monitoring, future research, we already did like a lot of efforts and a good job, but we should continue to conduct this high quality measurements in the future to monitor the ocean acidification and the anthropogenic carbon change. And that's it. So I'm happy to take any questions out there. Thank you.
Thanks, Xinyu. So if anybody has any questions, we'll take them after Wiley's presentation. But if you want to submit them in the chat now, that's fine. So next up, we'll be hearing from Wiley Evans. Wiley's a chemical oceanographer at the Hakai Institute in British Columbia, Canada. The Hakai Institute is a non-government agency that focuses on science along the coastal margin of the Northeast Pacific. Wiley's research focuses on understanding patterns in marine carbonate chemistry in coastal ocean settings, and he has nearly 2 decades of experience making underway CO2 measurements along the Northeast Pacific coastal margin. Wiley's talk will focus on using volunteer observing ships to monitor coastal CO2 in nearshore settings. He will include information from a recent publication, as well as highlight how volunteer observing ships should be viewed as part of an ocean observing portfolio, and that these platforms can be utilized to measure more than seawater PCO2 with standard ancillary variables. Go ahead, Wiley. You want to share your screen?
Sure. Thanks, Emily. Thank you all for letting me present today. Joining you from Campbell River on Vancouver Island. Happy to be here working and playing in unceded territories of the Likwita people. And I'm really happy to be talking to you today because I grew up in New England, so I feel like I'm speaking to folks from home, which is great. And so I'm just going to share my slides here. You're hopefully looking at the correct slide.
Yep, looks good.
Yep. Okay, cool. So I was given a bunch of questions, as I think Xinyu was, with regard to things you're thinking about for developing your observing plan. And obviously I'm going to talk about volunteer observing ships, but I feel like my guiding question that I'm going to be speaking to is how we can use volunteer observing ships to monitor climate and anthropogenic signals in nearshore and estuarine environments. People have been, measuring oceanographic variables from ships of opportunity for many decades, but I think what I'm presenting here is a little bit new in the sense that it's in a really nearshore setting. What you're looking at is a picture of the Seaspan Royal, a tugboat I'll get to at the end of my talk. You're also looking at Campbell River on the left where I am right now, and you're looking up Johnstone Strait, Discovery Passage towards Alaska. This is the Alaska Marine Highway, or Inside Passage. And so most of what I'm going to be talking about is data collected along this Inside Passage from a paper that was published last year. These data were collected from a ferry, the Alaska ferry, the Columbia, pictured there. And I'll just highlight that the Inside Passage is an area that is vulnerable to ocean acidification and is potentially a high risk region. We know this because we published a risk assessment in 2015 that was specific to the Alaska fisheries sector. And in this assessment, we consider risk as the intersection between hazard or ocean acidification, the presence of vulnerable species, and our dependence on those species. And so where all of these things converge, the boroughs in this map around the state of Alaska become red. And so you'll see Southeast Alaska is a region that is very high risk for ocean acidification. What's key from that analysis is that the hazard portion of it was based on models, and we don't have a lot of data in this area, or at least we didn't when we started this work. And so the picture on the right shows an image from the Surface Ocean CO2 Atlas. This is from 2019. So it's a few years old, but it's about the time we started. Well, we were underway at this point, but you can see what I'm showing is tracks from vessels making underway CO2 measurements, and the color is the month of the year. You can see there's barely any tracks in Southeast Alaska or down the Inside Passage into BC, and the tracks that are there are single month measurements. So we really didn't have a lot of information in this area. There was a few places where there's fixed time series. The one I'm highlighting there with the panel on the bottom is a NOAA buoy that was decommissioned in 2016. That gives us a nice picture of the seasonal pattern in sort of more exposed shelf setting in Southeast Alaska. There's a few other time series sites that are indicated by the stars, but generally we didn't have a lot of information about how marine CO2 system parameters varied in this area. And that gave me the arsenal I needed to approach the Tula Foundation, our parent foundation, and the Alaska Ocean Observing System to help fund the installation of an underway system on the Columbia. We also got some funding support from the Alaska Coastal Rainforest Center. And so with all of that funding support, we were able to install a system on the Columbia, and it was not a small feat. There was no seawater line to start with. And so that was quite a bit of engineering to get the line in there. But what you're looking at is the bow of the Columbia, and the cavity on the bow there, just below the water line, which is where the paint gets faded, is about 2 meters below the water line. That's the bow thruster cavity. In that cavity, seawater is drawn in to cool the bow thruster engines. So we tapped into that line, which is the picture on the bottom in the middle. That's the tap into that bow thruster cooling line. We make a temperature measurement right there. That's our in situ temperature measurement or a sea surface temperature measurement. And from there, water is pumped up to the car deck where we have a General Oceanics 8050 CO2 system, which is sort of the gold standard. It's highly autonomous, also highly expensive, but it's what's used mostly around the world and by the NOAA fleet. We're drawing in air as well for an atmospheric CO2 measurement. The air intake is on the foredeck, where we are also getting our position information to go along with all the data that we're collecting. The system is measuring atmospheric seawater PCO2, temperature, salinity, oxygen. And in 2019, we teamed up with Yui Takashita from Minbari, and he installed his BGC Sumo, which is a pH measuring system. Also, we had nitrate on there, but the nitrate channel didn't function very well on this ship. So we had corresponding pH data for the latter part of 2019. This is the transit of the Columbia. It's 1,600 kilometers, which we think is probably one of the longest instrumented ferry runs in the world. Runs from Bellingham, the B at the bottom of the map on the left, up to Southeast Alaska, basically along the Salish Sea, central north coast of BC, to Ketchikan, north to Wrangell and Petersburg, Juneau, up to Skagway, where the Gold Rush jump-off point was, Haines, Sitka, back to Ketchikan, and back to Bellingham. It does this whole circuit in a week. So it's just continually running the 1,600 kilometer circuit. The color on the left is salinity variability. And so that's coefficient of variation as a percentage. The reds are very high variations in salinity due to freshwater input. You can see there's spots along the transit where there's a lot of freshwater addition and high variability, but a ton of freshwater is coming in from Southeast Alaska, particularly in the northern end of our transit around Juneau and in the area of Lynn Canal up to Skagway. Area also has large phytoplankton or chlorophyll variability because of periods of high production. This is what the data set looks like. I have a number of these types of figures, so I'm just going to orient you to them quickly. You're looking at latitude versus longitude and time. It's a three-dimensional image. You can see the map sort of on the left side of the panel, of each panel, and the dots showing the terminals that I just described. And each line of color is a transit. There's 135 transits over this 2-year data set. Unfortunately, the ferry stopped running in 2019 and actually just came back online about a month ago due to COVID and budget issues in the state. And so we're looking at temperature and salinity. You can see the seasonal warming, but it's not uniform everywhere. So that's important to recognize that we have large warming in sort of protected inland waterways, but areas where there's tidal mixing all year, like the pinch point between Vancouver Island and the mainland, it never really warms up. We also have spatial pattern in the freshwater input that I was alluding to in the previous image. And you can see that big area of blue in the northern portion of the transit. That's the freshwater addition around Juneau and Lynn Canal. Here's the oxygen and PCO2 data from the ship. And I'm presenting the oxygen here as a difference from saturation, so a delta O2 term. It's not as pretty as oxygen argon because you can have bubbles and things in there that are impacting the excess oxygen and the deficits, but it's largely driven by production and respiration. So areas of warm colors are areas where there's probably a strong production signal, and that's supported by the lower panel where we have CO2 drawdown. And so you see these things mirroring each other. And it's a patchwork similar to the warming pattern that I was describing. Certain regions along the transit exhibit strong drawdown and high excess oxygen over the spring and summer. Areas where there's mixing do not. And so that's an important difference that we're seeing along this transit. We started to see some evidence of interannual variability in this 2-year record. Year 2 looked to have stronger undersaturation in PCO2, slightly higher excess oxygen. but it was really hard to say with just 2 years of data. There's a lot of variability here, and we want to try and explain some of this variability. And just for this group, I think it's important to recognize this equation, which I think is the most important equation to explain PCO2 variability. This comes from a paper by Taro Takahashi in 1993, sort of one of our godfathers for underway CO2 system measurements. And what this equation is saying is that variations in PCO2 are governed by variations in temperature, salinity, and TCO2, or total dissolved organic carbon, or DIC. I'll use TCO2 in this talk, and alkalinity. And how those terms vary, PCO2 is described by the arrows in this slide, but largely it's thermodynamic, so warming, freshwater addition, and biophysical terms, which is mostly net community production, physical transport, and mixing in this area. And so these terms all interact with each other and become more or less important over different time and spatial scales. And so over seasonal scales, we can look at the amplitudes of these individual terms and try and understand what's most important in this setting. And so that's what we did, was we looked at sort of the thermodynamics, the changes in temperature and salinity versus what's called the biophysics, and tried to decide what's more important, how that spatial variation is captured. And so what I'm showing you here is the ratio of the amplitudes of the specific component, temperature on the left, salinity in the middle, and the combination of temperature and salinity on the right versus the remaining components describing PCO2 variability. And from the left, you can see the temperature component is pretty minimal. There's certain areas where it's slightly higher because of warming, but generally it's quite lower compared to some of the areas where salinity is very important. As you can see in the middle panel, salinity variation dominates PCO2 variability in particular areas, specifically that region around Juneau and Lynn Canal, and some other areas around the Fraser and on the central and northern BC coast, where we have higher freshwater addition. The combination of temperature and salinity, I think, is important to think about because they can oppose each other. For instance, in Lynn Canal, that water is very cold. That's coming in, that glacial meltwater, and so that can oppose the salinity variation slightly. This is nice, but we don't want to just talk about PCO2 variability. We do want to get to pH and saturation state, and we largely only measured PCO2. So we needed an alkalinity-salinity relationship to use with our data to say something about these other terms and to evaluate a particular alkalinity-salinity relationship that we used. We collected discrete samples and compared those to what we would think the alkalinity would be based on those relationships. We did that on 2 cruises, one in November, which is the top set of panels, and again in August. And what you can see in the November data is there was great agreement, actually. The discrete data, which isn't directly measured alkalinity, I want to point out, we in our lab measure PCO2 and TCO2 and calculate alkalinity. And so one of our technicians, Katie Pocock, is doing our measurements there in that image. But the alkalinity data agreed really well with what you would expect it to be based on that relationship for November. And the panel, the second panel down, shows the difference with the gray horizontal lines as plus or minus the root mean square of that relationship. Conversely, in August, things diverged quite a lot, particularly in the northern part of the transit in that area of highest freshwater addition. We were very off by as much as 200 micromole per kilogram in those freshwater areas. We're overestimating alkalinity. With Wiley's system, we were able to look at this in a little bit more detail. What I'm showing you here is the salinity that corresponds, the salinity tracks that correspond with the pH data collected with Wiley's system. And the panels in the middle show the measured pH versus estimated from this regional alkalinity-salinity relationship, and it's colored by salinity. And so you can see all the data that's falling off the 1-to-1 line is blue. and it's all up in Lynn Canal. It's all in this area where we get this massive freshwater addition. Again, we're overestimating alkalinity, which will lead to an overestimate of saturation state in this region. And so that's something we recognize, but we understood the direction of the problem. So we decided to carry on with our analysis, recognizing that we could improve our constraint of saturation states in Lynn Canal in the future. And so this is what pH and saturation state data look like when we're applying our regional alkalinity salinity relationship, very similar to the oxygen PCO2 data that I showed before. What I want to highlight, though, with the saturation state is we're seeing undersaturation in some regions through the year. That area between Vancouver Island and the mainland, Johnstone Strait, Discovery Passage is corrosive to aragonite throughout the year. We're seeing areas that are corrosive over winter, Salish Sea, some of the Central Coast, Southeast Alaska, and then in Southeast Alaska during summer, in this area of high melt addition, low salinity, we're seeing very corrosive conditions despite low PCO2 and despite overestimating saturation state. So we know that we're overestimating alkalinity. It's still corrosive. It's probably more corrosive than we're estimating it here to be. We can look at sort of the variability in these terms and try to describe differences in terms of the severity and the timing, which is what we've done. So the top panels are pH and the bottom panels are saturation state. First, you're looking at the coefficient of variation. The severity, which is defined as the lower 5th percentile of the distribution of data collected over the 2 years, and the timing of those most severe conditions. And what I want to point out is that this mixing zone, again, Discovery Passage, Johnstone Strait, which is right out my window here, has the most severe pH conditions along the transit, and those occur in the fall. And in fact, these areas where we do see tidal mixing that's really persistent through the year, we have lowest pH and highest PCO2 conditions in the early fall in those areas. And conversely, saturation state, most severe conditions was up in Lynn Canal, despite our overestimation of saturation state in that area. And those severe conditions were in August. And so very different in some notable differences between pH and saturation state in terms of their severity and timing that are probably important to consider when we're thinking about species impacts along this transit line. One of the other points we tried to address is, can we use this information, this rich data set, to help us inform and optimize our observing system? And so to really address this question, we calculated time of detection, which is a term that tells you how long a time series needs to be for a secular trend to emerge from that record. And what I'm showing you here is sort of the noise in the PCO2 data. We've removed the seasonal cycle and calculated the standard deviation of the remaining anomalies, and that's what that plot is. And you can see when you remove that seasonal cycle, there's some areas where the variability is actually much lower than in other regions. And when you calculate time of detection from those data, you can see that you can actually have an acceptable amount of time for an observing program to try and constrain long-term change. For instance, in this high CO2 region, where it's fairly stable, we may only need about something between 10 and 20 years to observe a secular change, as opposed to just in the northern Salish Sea, a bit south of where I am, it's highly variable, and it's going to take many decades to see that long-term change. So we've already set up an observing station at this site, and that's what the picture on the right is. This is at a hatchery, Cermak hatchery here, that has a pier that jets directly out into Discovery Passage that we've started making pH and oxygen measurements at in 2020. But without long time series, we're really stuck with trying to calculate and estimate anthropogenic CO2 like Xinyu was doing in her previous presentation. And the approach that we use is much simpler than the EMLR approach. It's a delta TCO2 approach. It's a basically a simplification of the C-star method if you read all of these things. But few important points I think are worth mentioning. One is that anthropogenic CO2 calculations have been done for quite a while. They really started to emerge in the 70s, and they all sort of assume that you can extract the signal from your measured TCO2 data. So that's I think key. For this approach, what we're really assuming is that we're looking at changes in anthropogenic CO2, and we're not really considering, at least at this point, changes in temperature and salinity. And so that's, I think, something to recognize. We're also assuming that the disequilibria in TCO2 between what we're observing and what we might expect seawater TCO2 to be if it was in equilibrium with the atmosphere is not changing. So that's constant. And that is, I think, an important assumption to recognize. We calculate that disequilibria term with the equation that's shown there. And the key point of this equation that Xinyu also keyed in on was the age. So the age estimate, I think, is really important. Without it, you can radically overestimate TCO2. And for us, we estimated the age using the apparent oxygen utilization divided by an oxygen utilization rate that we took from the literature. And so that was a constant, definitely something we could improve on, especially in these nearshore settings where that number is highly uncertain. And with that age correction, we can calculate the TCO2 for any given year. And then the anthropogenic CO2 signal ends up being the TCO2 for that particular year minus a baseline, in our case, 1765. Few important points from the images on the right. One is the atmospheric CO2, and I just want to highlight the 1.5 C warming level. This corresponds to an atmospheric CO2 of about 470 ppm or microatmospheres. That'll come important in a minute. The star on that dot is basically following the shared socioeconomic pathways from the recent IPCC reports. And the bottom panel is the age. And so that's our age assessment for surface water along the transit. And you can see it varies spatially and temporally. This approach has an uncertainty of at least 5 micromole per kilogram, probably double that if you consider a 50% uncertainty in our age estimation, which leads to uncertainties in derived hydrogen ion of about 1 nanomole per kilogram, pH of about 0.03 or 10 times, the high quality pH measurements, and a saturation state uncertainty of about 0.08. Okay, so here's what the anthropogenic CO2 looks like for the transit when we apply that approach. It's quite variable, which is really interesting. Actually, the interesting parts are that the highest measured or highest determined anthropogenic CO2 agree with the open ocean assessments that are very seasonally biased. These are summer cruises, surface water, atmosphere, anthropogenic CO2 of around 60, 65 micromole per kilogram in the Northeast Pacific, and that's what we're seeing here. So that's good to see. It's the trends towards lower values that are interesting. In the mixing zones, we're mixing up deep water, the conditions drop. In the areas where we have freshwater input, the anthropogenic CO2 load also decreases. We can take this anthropogenic CO2 content and calculate when we first see saturation states below 1 in this region, and that's what the bottom panel is. The way to look and think about this panel is the value where you see deep blue. These areas have always been corrosive. When you remove the anthropogenic CO2 signal, they're still corrosive. So they're natural hotspots for corrosivity, which are interesting, I think, if you want to go and understand how species are thriving there, because they've been thriving there for over 250 years. Other regions have started to emerge more recently. The Salish Sea really became corrosive during winter, likely around 1950s. Southeast Alaska looks like its winter conditions trending towards corrosive levels really emerged in the early 2000s. And the rest of the transit, it may emerge much later. So there's some nice nice variability in that emergence along the transit. This is what the acidification signal looks like from the data. I'm showing percent change in hydrogen ion content. The top panel is the contemporary levels. On average, about a 40% increase in hydrogen ion content, which corresponds to a pH decrease of about 0.14, so slightly greater than the global average. Notable spatial and temporal variability. Most important, I think, is the seasonal differences. So we see greater acidification in the winter when that water is more weakly buffered. Now projecting out to that 1.5 C acidification level, when we hit that atmospheric CO2 of 470 ppm, which could be in 2035 under a high emission scenario, we add another 13% of acidity. And if we track that high emission scenario, that's 50% of what we've seen so far in the next 15 years, and that's quite scary. Two last slides here. One closing point. Hopefully I've convinced you that there's a high degree of value in thinking about these nearshore vessels for observing, but they're part of an observing portfolio. And I think that's some key points that emerged for me and Xinyu's closing points as well. You have to look at how all of these pieces fit together. And that's what we've done recently in British Columbia with the formation of our Ocean Acidification Hypoxia Action Plan, which I hope the government releases very soon. But the map shows the distribution of assets that we have ongoing right now in BC, with the ferry as the green track, but we have gliders and we have moorings and we have all these, other assets that are underway. So I think it's really important to think about these things collectively. And the volunteer observing ships are certainly part of the portfolio. And then lastly, back to the tugboat, I think we can really expand these platforms, and that's what we're trying to do. And I was really pushing to actually have some cool new data to show you guys. But we've installed a CO2 system on this tugboat. It's the super CO2 system from Sunburst. It's pictured there on the left. It's a fraction of the cost of the GEO system. And we've made some modifications, stealing some ideas from GEO. So I think we're on our, it's not the same quality of data, but it's certainly very good data. And we're also adding in TCO2 underway. And so for the combination of these two systems on this vessel, we can do it for the same cost as a GEO system, which I think is really, really important and valuable, particularly in these areas that we have a lot of freshwater input. And finally, the picture on the right just shows the distribution of SOCAT data in 2021. So you'll see the ferry track now, it's the yellow. We have 12 months of the year resolved in all those pixels. Lots of gaps still. The red tracks are the tugboat data we've already collected since June of last year. And so we're filling in those gaps with this new platform, and I will end there. Thank you so much.
Thanks, Wiley. That was great. I'm going to hand things over to Chris Hunt at UNH. He's also a member of the NECAN Steering Committee. So go ahead, Chris. I noticed that Dwight had put some questions in the chat, and I think maybe Sam has some questions, but Chris, if you want to take it away.
I'll lead off the questions and then we can turn to Dwight and Sam's next. But first, thank you both. Those talks were excellent. And I'm really glad that it worked out that the two of you were able to talk at the same time, because I feel like your materials were really complementary in some ways, but in other ways were kind of two sides of a coin, where Xinyu was working with sort of depth resolved cruises that happen, during the summer, during the same time of the year, every few years, whereas Wiley's looking at surface data that's collected at a high frequency, but kind of just in that surface domain. And so this is a very broad question, and I don't expect you to have a complete answer, but I'd like to get your thoughts as to, when you're building, like Wiley, like you said, when you're building a monitoring portfolio, how do you balance those two? That it's hard to do everything everywhere all at once, but we have technologies to do these sort of underway automated measurements, whereas you have to send people out on a ship to get the depth measurements. And I think that they're both important. And so I'd like to get any thoughts you have about how do you look at balancing out those two factors and those two possibilities?
Xinyu, I'll let you go first if you want to comment, and then I can follow.
I just have like a really quick comment. I would see it's really convenient, and we have those underway measurements on different vessels. But I would see the research vessels that's dedicated to the OA cruise. It's more like a pillar of the OA research, because it provides the highest quality of all the carbonate measurements. And actually these measurements can benefit to other underway or other measurements as a ground truthing or calibration. That's from my perspective.
I agree. I think you have to have that ground truthing. And, we're fortunate in BC that NOAA comes up here as part of the West Coast OA cruises and does Queen Charlotte Sound line and does a line off Vancouver Island. iOS is operating Line P and doing their ground truth measurements at climate quality. So we have a collection like that. But if we just relied on those, obviously we'd be blind for a lot of the year and for multiple years, because there's big gaps in there. So you have to fit these other opportunities in. And to me, that's what they are, is opportunities. Like, we're working with C-SPAN on the tugboat. We have no control on where the vessel goes. Yep. It's hard to actually get to the vessel to service it sometimes, because they don't ever want to stop. So so you kind of have to think about those constraints and sort of layer them in with the other aspects that you have more control over, like where dedicated cruise work is going to go, or where you're going to put a mooring, that kind of thing. And of course, build in models, because we can't measure everywhere. And so I think liaising with the modeling community is really key. In BC, we have a lot of work to do in that area. We're quite a bit behind, I think, with our forecasting capacity. And so making sure that modeling folks are always part of the discussion and, leaning into where we need to really focus, I think, is key too.
To follow up on that, do you think that working with modelers would provide a way to better constrain your TA estimations?
That's a tricky one. I think it's easy to say yes, but I'm not sure that's really the case. I mean, there's a bunch of high resolution FVCOM models in our region that are using watershed data, but I think they're getting better at being able to look at the distribution of freshwater input across the area that we're in, but not so much the character, if that makes sense. So those problems might not lend themselves to a modeling discussion just yet.
Awesome, thanks. I guess since I'm talking, Dwight, do you want to read your questions, or do you want me to read them?
Sure, I can read them. My apologies if there's any echo. So first off, two really fantastic talks. Really insightful. I've got a bazillion questions for both of you, but I'll try to limit them. So I'll start with Xinyu real fast. I'll just go through the questions. So the derivation of anthropogenic CO2 that you utilize, I might be completely wrong, but I think there's a dependency or on some assumptions regarding residence time of the water masses. Does that factor in? And if it does factor in, I think we've had conversations with Brendan Carter about what it would require to better constrain that in the context of the COA cruises or alternate supplemental exercises, and maybe it doesn't need to be done every cruise. Maybe it's just something we need like a targeted study on. So what are your thoughts on how important the residence time estimate?
So I think the residence time estimation is really important. And actually in my studies, I got those informations basically from previous models, such as the models in the Chesapeake Bay or the Delaware Bay, and from the physical oceanography literatures, and to incorporate that residence time to my own box models to get it. So the influences of the residence time is mainly on the air-sea gas equilibrium. So if we have a very short residence time of the water, and maybe it's not able to get the air-sea gas equilibrium for that surface water. So in that case, actually, in some like very nearshore regions, the consumption we are using to calculate anthropogenic carbon is not that valid anymore. As for how to constrain them in the, I feel it's kind of hard for me to imagine what we can do from the observational part. Maybe someone can add something. But I would definitely suggest to try to incorporate my method to the model results and to validate. And at least we can get an uncertainty of my method, because right now we don't really have the way to get an uncertainty estimation of this very nearshore part.
So if the nearshore is the greatest area of uncertainty, I know like coral reef systems, when we used to try to do residence time determinations, there were some approaches whereby you look at an episodic event like a major rain event, and you look at the salinity recovery time as a way of kind of back calculating the residence time. So I don't know if there's like existing copious amounts of salinity data in these coastal domains obtained. I mean, certainly Wiley would be having it on his cruises, where maybe we can interrogate them for perturbation events like major rain events, and maybe get an estimate of you know, how quickly the system recovers. So that might be a proxy for that. So the other question I have is, you know that we lack the data in the Gulf of Maine. Is that just because the earlier GOMEC cruises didn't have the fidelity that ECOA has now? And now it's just a question of just making sure we're maintaining the high fidelity ECOA within the Gulf of Maine. Is that correct?
Yes, I think so. Yeah, because the GOMEC cruises, we only have one transect near the New Hampshire, and that's it. And this is not enough to estimate the whole anthropogenic carbon for the Gulf of Maine region.
There you go, Rick. Oh, I just muted. Okay, so third question is, so you mentioned the dissolved organic carbon was being collected, which I didn't even know. I was on the cruise. I didn't know we were doing DOC samples. How is that being post-cruise analyzed? And where do you see that it applies to the work you're doing, if at all?
This is actually, I'm not really sure about that too, because I'm not like really using the data, but I did see people collecting. I feel like Liza may do part of the work during the ECOA too, maybe. And I think that is important because anthropogenic carbon is not only stored in the seawater, and some may redistribute it to the DOC part, and some may just like sink to the bottom as part of the POC. And so I would say my estimations on the anthropogenic carbon in the dissolved organic carbon form is only part of it. So if we want to do a more fully comprehensive carbon budget on the anthropogenic carbon, we should see those changes too.
All right, great, thanks. I'm going to switch over to Wiley. Thank you so much for that fantastic talk, really helpful. And Wiley, really great stuff that you're working on. I'm wondering, and maybe this isn't just to you, but to everybody on the call, but do we know of an analogous coastal vessel that would provide an equivalent data set in the Gulf of Maine region? I mean, the coastal complexity that Wiley showed is very similar, particularly when you get up in Maine. And I'm not aware of anything like that. Is somebody?
Dwight, we had a project at one point where one of our objectives was to put monitoring assets on a cargo ship that ran a transect between Portland, Maine, Reykjavik. But it never ended up happening for a number of reasons. And then that ship was because AOML was operating a CO2 system on that ship, but it ended up getting pulled off. And so we had to change our plans. So there are ships like that do provide opportunities, but I'm sure, as Wiley can tell you, those also require a lot of manpower or person power to maintain and keep going, especially on private vessels where access might be limited when they're out at sea so much. And that ship also, like, it would go back and forth to Portland, Reykjavik, and then sometimes they would decide to go trundle around Europe for a month or two. And so it was a little bit less predictable in terms of where they went. But yeah, Wiley, you can speak a little bit to that too, I'm sure.
I know that AOML and POML, P and EL all sort of rely on the container ship fleet for these types of observations. And I think they're really of high value for open ocean crossings, right? But if we're talking about coastal, I would sort of consider looking around at other state ferries, for instance. Washington State operates a system for, I forget who is the science lead there, but they do have a system on one of the Washington State ferries. BC Ferries has got instrumentation from Ocean Networks Canada. We've done a glacier tour vessel in Prince William Sound in 2014 as well. That was a very small boat, and that worked out great. So it could be. It could surprise you. And I guess the other thought that I had was something that we're trying to get to with the tugboat we're doing now. We have access to that system every time it's in cell range. So I can log in and control the system and control the computer. We have data coming into our ERDAP server in real time. And right now we're trying to think about what more can we do with those data. Can we get them into a data portal that the public can look at and follow along as an outreach tool? And maybe there's even a science that can be done, sort of ingesting immediately into modeling for ground truthing, that sort of thing. I think there's a lot of potential there. And so having that live link to these nearshore vessels they're within range, they're in cell range all the time. You don't have to use iridium. In fact, don't, because it's super expensive. But yeah.
Okay, that's super helpful. And in fact, yeah, OAP funds a lot of those, like the Skogafoss and all others. And there's this division between GOMO and OAP, and we're trying to figure out where the lanes divide. And our focus has been the coastal large marine ecosystems on the OAP side, and the global data sets are really GOMOs. And I'm beginning to wonder, looking at Wiley's data set and stuff, if maybe OAP really needs to be pulling away from these large open ocean underway systems, leave that to GOMO, and that we should be looking a lot more closely at these ferries and very near shore coastal dynamic systems. So anyway, really helpful. Thank you so much.
Sam, did you have a question from earlier?
I did. Thanks to you both. Good to see you virtually, both of you, for your time and for your great talks, like Dwight said. I guess I was wondering, I have one question for Xinyu and then one for both of you. I guess, so Wiley kind of he said it, he didn't show us, but he told us that the numbers were similar to the open ocean values that had been collected, like off the, I believe him, but they were on par with that. Do you have a sense if the numbers that you've calculated in your work are sort of similar to the values from the like A16 or kind of the North Atlantic Basin? And then I kind of wanted to ask you if you could clarify some of the changes you reported, like the pH and whatnot. Were you talking about subsurface or surface, or kind of where in the water column? So if you could clarify that and then answer the other one, that would be great.
Yeah, so all the changes is the whole water column, but actually the surface is only for the Gulf Stream, right? Because the AIW North Atlantic deep water, they're the deep waters. So for the numbers, yes, it is comparable to the A16 changes, anthropogenic carbon changes. But there are several points that we noticed that is different from the coastal, the open ocean estimations. The first one is the sandwich structure. I also talked to Brandon about this. His new analysis didn't really show a sandwich structure, and that's what I'm thinking. That's because of the Labrador seawater actually reached the North Atlantic edge, mid-Atlantic bight edge first, and then spread to the whole Atlantic Ocean. So generally, the upper North Atlantic deep water in the whole Atlantic is still low in anthropogenic carbon, just only because of the topography that we see a very relatively high deep water anthropogenic carbon signal in the mid-Atlantic bight here. And I believe it can be found in the Gulf of Maine and even a little bit further north too. And also another information I got is actually I compared to the Waikoa data about anthropogenic carbon, and the East Coast is like a little bit higher than the Waikoa ones. I think that's mainly because of the water differences. Yeah.
Cool. And then the other question I had was about the maybe more coming back to Dwight's question about residence time, but kind of coming back to Wiley's comments about age. Xinyu, you were able to answer and give us your ideas about kind of how we could augment observing to kind of better constrain that aspect. But I didn't hear a response, I don't think, from Wiley on that. I would love to know if Wiley has ideas about some other ways that you could augment observing to help better constrain that bit.
Thanks, Sam. I just think in general, we suffer from a lack of rate measurements. And the particular rate that I'd love to see is oxygen utilization rate. And, the value that we pulled from the literature was from Dick's 2004 paper. And it was, I think, an average of the upper 100 meters for Northeast Pacific during summer. I mean, it was probably weakly at best representative of the setting that we applied it to. And it made me uncomfortable doing it, but we needed to do something, right? It was a starting point. That's something I would love to see improved on. I think it gets to the residence time question a little bit because they're related, right? Like if you have a long residence time, long age, and so and particularly in these settings like where we have complicated coastline, like we do in BC, we have fjords, we have longer residence time over sills and deep water, we have the enclosed Salish Sea. Knowing more about the oxygen utilization rates, I think, would really inform a lot of the stuff that we're talking about.
Cool, thanks. And then I have one other question for Xinyu, if I have time. Do I have time, or do we have other questions? I think Beth had a question in the chat too. So just quickly, you noticed there was a pH decline, from 2012 to 2018, but in the Mid-Atlantic Bight, and I don't remember exactly, but I thought the surface mooring observations from the Mid-Atlantic Bight showed no change, or maybe an increase. Maybe that saturation state. I was looking quick to go back. I don't know if somebody else remembers. Is that because you were looking more subsurface? Like, does that suggest that's the difference between a surface and the subsurface there?
Yeah, I'll get in very quickly. This is actually a chapter of my dissertation. Great to the point. So that the control of the pH surface is actually is more balanced by different process. So the air-sea gas exchange is balanced by there's some thermodynamic change. So if I calculate equilibrated pH that using the total alkalinity and atmospheric PCO2, actually it is featureless. So actually the surface pattern and the surface variations of the pH is mainly driven by the biological process. So on the surface, the biological signals actually covers the change more. And even if we see a profile, and we always see a high pH layer on the surface, and we begin to see the low pH, near shore low pH to the offshore high pH gradients in the subsurface. we cannot observe them on the surface. So I think that's mainly explain why from the surface observations and data, it's very hard to track the pH changes.
That's really important. Thank you so much. Beth, didn't you have a question in the chat? Sorry, Chris, I didn't mean to.
No, I was going to ask Beth if she wanted to present her question.
Yeah, thanks. So this is mostly for Xinyu, but it's following up on Wiley's discussion of the development of the OA hypoxia plan. So our ultimate goal is to get to that sort of plan here in the Northeast. And I'm wondering from Wiley, how did you develop that plan? And from Xinyu, what are the important things that we need to have in our plan in order to augment the ECOA cruises, assuming those are going to go forward into the future?
I will quickly add, except the ECOA, I would say because ECOA is always happening in the summer, even though it's like a very comprehensive and high quality. So have a larger seasonality coverage is very important to quantify that. And to be honest, this should be also a part of my maybe bias on uncertainty in my anthropogenic carbon estimation. And especially in the winter, because the sea surface temperature is lower than the summer, so we are supposed to have more CO2 absorbed in the water. So the anthropogenic carbon may be like a fluctuation. But what I do is just estimate of mean status. And this can be applied by a lot of autonomous platforms and other strategies, I believe.
And Beth, how we got started was we had the Ministry of Food and Agriculture here in British Columbia. They decided that they wanted to do a, how did they put it? Not so much an action plan, but an assessment of where we're at with ocean acidification observing. And they reached out to me and I said, no, you should do an action plan. And they at first they wanted to talk very specifically about the fisheries sector. And I said, no, we got to think broader. We have, First Nations communities here that are doing substantial subsistence fishing. We have dependent coastal communities that are, reliant on tourism. And there's much more than the fisheries sector, even though the fisheries sector in BC is large. And so it grew up from that, and it all happened in COVID. So we did a workshop series to engage the science community, the public, the coastal First Nations and other stakeholder communities, and the policy folks, and then pulled everything together and wrote an action plan and a scientific assessment. We were sort of mirroring what Washington State did, and that's been submitted to government about a month ago, and they hopefully will be releasing sometime very soon.
So they're not unlike this process that we're going through in doing the webinar series and then having a culminating workshop and coming up with something at the end.
You guys just get to meet in person, so that's great.
All right. Well, I think that at least personally, I could sit here and talk for several more hours with both of you. But I also don't want to monopolize your time. So I'm going to say thank you so much to both Xinyu and Wiley for your time and your expertise in helping us formulate our own plan.